Okay, well, I have to redo this video because I was nearing the end and then I got the blue screen of death, which I didn't know was a thing anymore, but apparently it is. So, F's in the chats, boys. I'll drink to that. So, what I want to do, again, because it screwed up on me, I want to go over a few papers. I want to go over this increased immune complexes of hypercretin autoantibodies in narcolepsy. And I think that there are some really cool, what would you call them, just thought experiments that can be gleaned from this particular paper. And then I also want to go over this one called autoantibodies reacting with vasopressin and oxytocin in relation to cortisol secretion. And perhaps if we have time, I'll go over this diagnosis of Alzheimer's disease based on disease-specific autoantibody profiles in human sera. So, I have to go over all of this again, but it's all right. It is what it is. So I'm not actually going to go over the entire papers. I'm just going to go over the background, the methodology, the principal findings, conclusion, and then I'll go into the discussion as well. So let's begin. So, I should also say, the reason why I want to go over this is because we do have to worry about how viruses can frequently cause autoimmune diseases by the mimicry of your particular peptides. But that is just the first step that could happen. There is another possibility that if you do end up creating an autoantibody against a peptide, your own body can make an antibody against that autoantibody. And so you can end up creating complexes of these antibodies, and those can actually muddy the waters as far as when we try to do experiments on autoantibodies. So sometimes your body fights back, right? Sometimes you create an autoimmune disorder and then your body fights back against it with another antibody. So it's fighting fire with fire, if you will. But the problem with fighting fire with fire is that things can get burned. So if you make another antibody that can bind to the first autoantibody, that second antibody might possibly be able to behave similarly to the original peptide that you made the first antibody against. And I'll go over it later, but it opens up the door for more dysregulation down the road. And why do we need to worry about this? Well, mankind is currently being exposed to a very large number of a new disease that we've never experienced before. And the problem with this is that think about this with me. As you go through your life, you are going to be infected with new viruses, new pathogens, new everything, and new microbes, new fungi, and you are going to be making antibodies against those things. And with every one of those things, there is a small chance, a small opportunity that they are going to actually have a peptide that looks similar to one of your own amino acid sequences. And if that ends up happening, you are at least starting or kindling up a new antibody disorder. And we would hope that it never comes to that. We would hope that you might form an antibody that is slightly autoreactive, but you would hope that it dies down after the pathogen is left. There are examples where this doesn't happen, for instance, with, say, celiac disease. The only thing that really ramps it down is if you completely cut out gluten from your diet, right? So, but where does this, and we could also say that perhaps the older and older you get, Murphy's Law comes into effect where you've given your body enough chances to screw it up, therefore you end up screwing it up later, or you end up creating so many of these autoantibodies that might be associated with aging, that might be associated with having worse clinical outcomes the older you get. So, I mean, this might be one of the reasons why your immune system tries to lower in efficacy the older you get, because you probably have all these autoantibodies that would end up destroying your own body if your immune system didn't weaken. So this is just something to consider and think about. So what I want to do is go over this paper to highlight the way that autoantibodies can cause certain clinical disorders. And the first one is I'm going to go over is narcolepsy, and narcolepsy with and without cataplexy. So first, increased immune complexes of hypocretin. And what is hypocretin? This is one of the peptide hormones that your body produces. So let's go to Wikipedia because I love Wikipedia. So So hypocretin is also called orexin. So this is unfortunately another noisy part of biology and biochemistry, is we frequently will have multiple names for the same thing, and that kind of pisses me off too. But let's go through it anyway. So orexin, also known as hypocretin, is a neuropeptide, and it regulates arousal, wakefulness, and appetite. The least common form of narcolepsy, which is type 1, in which the sufferer experiences brief losses of muscle tone, and this is called cataplexy. So this would be a form of narcolepsy with cataplexy. This is caused by a lack of orexin in the brain, and this is due to the destruction of the cells that produce orexin. So if you can imagine, if you can kill the cells in the brain that produce orexin or hypocretin, then you will end up having narcolepsy with cataplexy. Okay, and cataplexy is just generalized fast-acting muscle weakness that happens almost randomly. So let us continue with our paper. So, and remember, autoantibodies are antibodies that are attacking something within your own body. So it's an autoimmune disorder. Background. Hypocretin peptides participate in the regulation of the sleep-wake cycle, while deficiency in hypocretin signaling and loss of hypocretin neurons are causative for narcolepsy cataplexy. So losing these hypocretin neurons or deficiencies in hypocretin signaling, either of those can cause narcolepsy with cataplexy, and that is called NC in this paper. However, the mechanism responsible for alterations of the hypocretin system in narcolepsy cataplexy and its relevance to other central hypersomnias remain unknown. So hypersomnia is when you need to sleep way too much. So here we studied whether central hypersomnias can be associated with autoantibodies reacting with hypocretin 1 peptide present as immune complexes. So an immune complex is when antibodies are not free. They are binding with a bunch of other things to create larger structures. So those are immune complexes. And one of the downsides of an immune complex is that sometimes these do not show up in antibody tests. So certain antibody tests will not show them because they're complex, they're used up, so they won't bind with whatever test you are using. So an immune complex is sometimes called an antigen-antibody complex, and it is a molecule formed from the binding of multiple antigens to antibodies. The bound antigen and antibody act as a unitary object, effectively an antigen of its own with a specific epitope. So here they're talking about immune complexes where you have an antigen, so you could view that as a protein that a virus has made or a bacteria has made, and the protein will have multiple sites on the protein that 2 different or more antibodies can bind to. And when this happens, or you could also say that it is possible that the protein is symmetric too. So one protein might be able to have the same antibody bind in different areas. But what can happen here is that you can form chains or clusters of antibodies and antigen. So that would be a type of immune complex. So those are one type. Another thing that can cause immune complexes is if you have anti-idiopathic antibodies. I will call them anti-antibodies because they are basically, you have an antibody and then you have another antibody that your own body produces, and they lock together perfectly. So your body can sometimes make an antibody that neutralizes a different antibody. Okay, and that might actually be beneficial. That might actually be part of a regulatory mechanism to prevent autoimmune diseases. But um, who knows? It could actually be completely random and not intended at all. So we'll go over this paper as quickly as possible because I wanted this to be done quickly because I worked today, and so I wanted to get this done quickly, but apparently that's not going to happen. So the methodology. Wait, yeah, serum levels of free and dissociated, which are total autoantibodies reacting with hypercretin 1 peptide, were measured by enzyme-linked immunoadsorbent assay, or ELISA. And these were analyzed with regard to clinical parameters in 82 subjects with narcolepsy, cataplexy, narcolepsy without cataplexy, or idiopathic hypersomnia. So idiopathic means without a known cause. So, and this was compared to 25 healthy individuals. So the principal findings was that serum levels of total, but not free immunoglobulin G autoantibodies against hypercretin 1, these were increased in narcolepsy cataplexy. So not the free immunoglobulin. So remember, IgGs, those are one of the long-lasting immune antibodies that your body produces. Serum levels of the total amount, so that is including the IgGs that are in these larger immunocomplexes that are used up, if you will. Those are in higher concentrations in narcolepsy, cataplexy, but the free ones that are just floating around waiting to find something, those are not increased. So, and we'll go into detail of what this could possibly mean, what mechanism would cause this. So anyways, increased levels of complex IgG autoantibodies against hypercretin 1 were found in all patients groups, with a further increase in narcolepsy cataplexy. So the autoantibodies that are complexed in these large groups, those are elevated in all of the patients with these narcolepsy or narcolepsy-like disorders in comparison to the control group. So increased levels of complex immunoglobulin G autoantibodies against hypercretin 1. Oh, I should also say it was even higher in narcolepsy cataplexy than the other groups. So it appears as though the more of them that you have, the more severe your clinical diagnosis will be. So there is a spectrum to it. Anyways, levels of total IgM, hypercretin autoantibodies, were also elevated in all groups of patients. So IgM is the short term or the short acting antibody. It's produced, it's one of the first antibodies to be produced as a result of an infection, or as of the result of an antigen being presented. So this would indicate that whatever the body's experiencing to cause this narcolepsy or to be associated with this narcolepsy, it doesn't seem to just be a chronic thing. It seems to be a fast acting thing as well. It seems the immune system is activating over and over again and to produce these antibodies against something within the own body or within your own body. Anyways, increased levels of anti-idiotypic IgM autoantibodies reacting with hypercretin 1 IgG autoantibodies affinity purified from sera of subjects with narcolepsy cataplexy were found in all three groups of patients. So, so the levels of total IgM hypercretin 1 autoantibodies were also, oh yeah, wait. So here we're getting a little bit more complex. So increased levels of anti-idiotypic IgM autoantibodies. So let's say you have IgM autoantibodies. You can also have anti-idiotypic or anti-antibodies. So you have an antibody that this is the IgM that is attacking the hypercretin 1. Well, you can have another antibody that is going to attack the first one. So this would be anti-idiotypic. So let me look this up to make sure that I'm giving you the right information. So, well, here they're doing it in reference to vaccines, but we'll just go along with this because it teaches you the same thing. So anti-idiotypic vaccines comprise antibodies that have three-dimensional immunogenic regions designated idiotopes that consist of protein sequences that bind to cell receptors. So idiotopes are aggregated into idiotypes specific to their target antigen. An example of an anti-idiotype antibody is raclutumab antibody. Raclutumab antibody. Let me look up anti-idiopathic definition. So I might have to go into a we'll go into this anti-idiotypic antibody. So an anti-idiotypic antibody binds to the idiotype of another antibody. So you have the idiotype of your first antibody, which can bind to an antigen. So an anti-idiotype antibody will also bind to that first antibody. So that's why it's called anti-idiotype antibody. Now, can you see a problem with having an anti-idiotype antibody? I can. So here, this area of this anti-idiotype antibody might look very similar to the antigen. So this, let's say this isn't an antigen. Let's say this is an autoimmune disease. And this is actually a peptide in the body, a peptide hormone. So this antibody has, let's say this is a key. This is a key that fits into a receptor, and that receptor is a lock. So we can say that for, what was it? What is the drug we are? Hypercretin. So let's say we're talking about hypercretin or orexin. Let's say this is hypercretin or orexin, and you have an autoantibody that attacks hypercretin or orexin. Well, the hypercretin is the key, and you have a receptor in the body that is the lock that accepts the hypercretin. This antibody binds to the antigen. So this is almost like if you take a clay tablet and you press the key into it, you can create a new, you can create an indentation of that key. So what happens if you take that clay tablet that has the indentation of this key and you take another clay tablet and take an imprint of that imprint? Well, you are recreating that key. So this is how you can recreate a key. You take an imprint of the key and then you take an imprint of the imprint and you can recreate the original key. So here an anti-idiotypic antibody might actually function or look similar to the original peptide hormone. So this would possibly, as a result, mean that this is a new key. It might not work the same way as the original key, but it might be able to fit inside of the original receptor. So remember, you have orexin that can go into a receptor. This antibody, which looks like orexin, might be able to interact with that same receptor. So this might be a way at which full-fledged autoimmune diseases progress worse, or I should say devolve or become worse and worse over time. So that is what an anti-idiotypic IgM autoantibody is. Let's see. Increased levels of anti-idiotypic IgM autoantibodies reacting with hypercretin 1 IgG autoantibody affinity purified from sera of subjects with narcolepsy cataplexy were found in all three groups of patients. So all three groups not only had elevated levels of autoantibody, they had elevated levels of anti-autoantibody. So disease duration correlated negatively with serum levels of hypercretin 1, IgG, and IgM autoantibodies, and with anti-idiotypic IgM autoantibodies. So disease duration correlated negatively with serum levels of hypocretin 1. Okay, so here's the conclusion. Central hypersomnias, and particularly narcolepsy with cataplexy, are characterized by higher serum levels of autoantibodies directed against hypocretin 1. And these are present as immune complexes, and this immune complex is most likely with anti-idiotypic autoantibodies. And this is suggesting their relevance to the mechanism of the sleep-wake cycle regulation. So perhaps these autoantibodies and anti-autoantibodies play a normal role in they might play a normal role in your sleep-wake cycle, but it's the dysregulation. It is the overexpression of these things that end up causing disease progression. So I am not going to go through the actual paper. I'm just going to go into the discussion later. If you want to read this paper yourself, I'm going to leave a citation in the description down below. So, discussion. So, the present data revealed altered characteristics of hypocretin 1 reactive autoantibodies in subjects with central hypersomnia. We found that levels of hypocretin 1 free immunoglobulin G autoantibodies were lower in narcolepsy with cataplexy, narcolepsy without cataplexy, and the hypersomnia idiopathic groups of patients than in controls. So the free immunoglobulin Gs, the autoantibodies, those were lower in those with disease. So these data are in agreement with a previous report showing low levels of free hypocretin autoantibodies in narcolepsy patients. But because a fraction of autoantibodies exist in immune complexes, which are not detectable in normal assays or normal tests, we measured the level of total autoantibodies reactive with hypocretin 1 peptides using a sodium chloride glycine buffer, so which dissociates immune complexes. So they didn't want to just test the free antibodies. They wanted to test all of the antibodies, including the ones in immune complexes, and sometimes using salt solutions and using specific types of buffers, those can dissociate or destroy those complexes, and then you can run the same test on those destroyed complexes to see whether or not you had the antibody that you wanted. Anyways, with this approach, we found that in contrast to the free fraction of hypocretin 1 autoantibodies, the total immunoglobulin G, which is the long-lasting antibody, IgG autoantibodies are increased in patients with narcolepsy, with cataplexy. So we acknowledge some limitations of this analysis since few values of total IgG autoantibodies had optical densities above 2.5. So this, which might not be very accurate and could underestimate the actual autoantibody levels. So here, when we do a lot of our quantification of concentrations, we very frequently will use standard curves, which a standard curve is when you basically have a standard. So if you have a bunch of standards with known concentrations and you generate a line that is associated with a signal, and you can basically have a signal to concentration ratio, and you can use that ratio to know your concentrations. But the problem is sometimes if you over extrapolate past your standard curve, sometimes there can be the error can be way too big. So it's very important not to over extrapolate. And the problem with optical density measurements is that if the optical density, which is a measurement of how concentrated your protein solutions are, if those are too high, you might actually be underestimating the true number of the true amount of concentration in your solution. So here they're acknowledging that there might actually be an even stronger correlation because the optical density was far greater, that was above 2.5. which apparently is probably one of the standards in this particular test. Anyways, furthermore, by analyzing the ratios between total and free autoantibody levels, which shows proportion of complex autoantibodies, we found that all three groups of patients, including narcolepsy with cataplexy, narcolepsy without cataplexy, and hyperinsomnia, they all had higher percentage of hypercretin 1 autoantibodies present in immune complexes than in controls. So more of the antibodies that are against orexin and that against hypercretin, more of those, a larger percentage of them, are complexed in some way. So what can cause this? So in addition, using hypercretin 1 reactive immunoglobulin autoantibodies, affinity purified from serosamples of narcolepsy with cataplexy patients, we detected a band that corresponds to hypercretin precursor protein in rat hypothalamic homogenate in Western blotting. In addition to using yeah, I didn't exactly get this, and I don't think it's that important. Anyways, increased levels of immune complexes of hypercretin 1 immunoglobulin G autoantibodies, especially reported in narcolepsy with cataplexy, may signify deficient hypercretin signaling, as has been shown recently for alpha melanocyte stimulating hormone autoantibodies. So this alpha melanocyte stimulating hormone, or alpha MSH, this might be of importance for us as well, because this, remember, I had another video on this. There is a protein called POMC, the pro-opiomelanocortin protein. This is a protein that is synthesized and then has a bunch of different hormone peptides that are within it. So what happens is it is synthesized as the whole, and then peptidases or proteases will come in and break up the protein into its individual peptides, its individual signaling hormones, and then those are moved and categorized so that they can be used for cellular signaling when the need arises. So alpha melanocyte stimulating hormone, and I'm going to search this up just to make sure that I'm not POMC. Yeah, so it is derived from POMC. So POMC doesn't just have alpha melanocyte stimulating hormone. One of the other peptides in POMC is corticotropin. And we know for a fact that influenza and coronaviruses, and possibly countless others, but we haven't proved that correlation yet. We know that in those two subsets of viruses that they can create, or I should say, they can make your body create autoantibodies against corticotropin, or ATCH, which is a part of POMC, which is the same protein that this alpha melanocyte stimulating hormone is from. Okay, so, and apparently autoantibodies against alpha MSH is also implicated in clinical diseases. It seems like all these peptide hormones are vulnerable, if you will. So my concern is that viruses have already learned to alter your metabolism or alter your cellular signaling for corticotropin in order to survive and then make you more infectious. Is it possible they've done it with a lot of other peptide hormones? And I'm going to err on the side of yes, they probably have learned how to induce a lot of these other very serious diseases. So, and we'll keep on going, and then I'll say what I have to say later. So, however, the relevance of systemic hypercretin to the central hypercretin signaling is currently unknown. Peripheral sources of hypercretin, even subject to controversy, may exist. So there might be other places that are producing hypercretin, wait. So the central hypercretin signaling is currently unknown. So there might be some peripheral non-neuronal sources of hypercretin. Um, but we will ignore this for now. Whether hypercretin reactive autoantibodies are functionally significant remain to be determined, as most of immunoglobulins do not cross the blood-brain barrier under physiological conditions. So this is one of the issues that we have, is that how are these autoantibodies doing anything to these peptides if the peptides are within the brain, but the antibodies can't get to the brain, at least they can't get to the brain easily. So this is something that is of concern and might be one of the reasons why the central nervous system is considered an immunoprivileged site. So perhaps there's a lot of suppression of the immune system in the brain, possibly due to these autoantibodies that can cause significant clinical features. So continuing, recent study reports altered levels of main subclass of total IgG in NC and HI patients, which further support the involvement of the humoral immune response in central hypersomnia. Anyways, the composition of immune complexes of hypercretin 1 autoantibodies is most likely with anti-idiotypic autoantibodies. So these are the anti-antibodies or the anti-autoantibodies. And since increased levels of such autoantibodies was found in all three groups of patients with central hypersomnia, and they're presuming that the autoantibodies are being sequestered into larger complexes with the anti-idiotypic autoantibodies. Although we checked for the IgM class of antibody or anti-idiotypic autoantibodies, it is probable that other immunoglobulin classes, including IgG, may form such complexes. So they were only testing for the IgM anti-idiotypic autoantibodies, but it's possible that there might have been an IgG version as well. Anyways, the presence of anti-idiotypic autoantibodies per se is a natural phenomenon constituting the anti-idiotypic network, which role is believed to prevent autoimmunity through neutralization of persistent antibodies to endogenous or exogenous antigens. So this is a normal response, possibly, to create anti-idiotypic autoantibodies might be a way at which people avoid autoimmune diseases. And if it runs correctly, it might be a very good way to help avoid autoimmune diseases. But there might be issues when that becomes dysregulated. And it appears as though in narcolepsy cataplexy, that is the case. So, thus, our findings of elevated levels of anti-idiotypic autoantibodies in patients with central hypersomnia may be biologically significant in controlling the peripheral hypercretin availability. So they're saying there might be a balancing act between the anti-idiotypic and thus our findings of elevated levels of anti-idiotypic autoantibodies in patients with central hypersomnia, this might be biologically significant in controlling the peripheral hypercretin availability. So perhaps the production of more of these anti-idiotypic autoantibodies is allowing for a larger production of hypercretin. Granted, it might not be enough, but it might not be enough to prevent clinical problems, but maybe it'd be even worse without it. So nevertheless, considering the eventual complementarities between the paratope of anti-idiotypic autoantibodies and the antigen, so this, which is in the case of hypercretin 1 peptide, or a cross-reacting protein, one cannot exclude binding of these autoantibodies to the hypercretin receptors. So this is something that I'm concerned about with hypercretin, is that if you make an anti-idiotypic autoantibody, that anti-idiotypic autoantibody might look similar to the original peptide. And this is where you can have a problem, because that means there is a possibility that that second antibody is able to bind to the receptor the same receptor that the hypocretin can bind to. And it's not just for hypocretin, it could be for any peptide, any peptide hormone that you have both an autoantibody and an autoantibody that, or where you have an autoantibody and an anti-autoantibody. That anti-autoantibody might be able to bind to the same receptor. And so there is a definite problem there, because it would be possible that autoantibody could activate that receptor, it could block that receptor, it could cause an immune response and kill those cells as well. So all of those things could result in some clinically significant problems. So for instance, if it can bind the receptor and activate it, maybe that's a good thing because it can make up for some of the lost peptide hormone. But what if it binds and doesn't do as good of a job? Or what if it binds and blocks it? Well, then you have a problem there because it's an antagonist. And then not only are you losing your peptide hormone to the autoantibodies, but then you're losing the receptor to the other autoantibodies, and then both of those could make the disease far worse. Okay, so that is something that needs to be considered as well. And it could be, you would hope that the antibodies cancel each other out, but that's not what happens. Biology is a noisy field. The antibodies might not be canceling out. They might still be attacking their own, one might be attacking the peptide and one might be attacking the receptor. And frankly, it's nearly impossible to know what exactly is happening in the body for all of the different chemicals that's floating around in it with all the different receptors and peptides. So yeah, this is a concern. So such a phenomenon was shown, for instance, for the anti-idiotypic autoantibodies against anti-beta endorphin autoantibodies being able to antagonize. So antagonize means that they bind and then they basically prevent it from working properly. So those anti-beta endorphin autoantibodies can have an anti-idiotypic autoantibodies and that can bind to the beta endorphin or that can bind to opiate receptors. So remember, when you take opiates or opiate drugs, those are working on the same receptors that endorphin is working on, which is why when you have an endorphin rush, it feels so good, or why when you take an opiate, it feels so good. And don't take drug, kids, but just so you know, drugs feel good, which is why they're addicting, which is why you shouldn't have an unhealthy relationship with them, because it feels too good, and therefore it can screw you up. Regardless, pushing that to the side, there is, in one case of major depression, and I'm actually going to search this up so that I do a paper on it later, or do a video on it later. Not only was someone making antibodies that would just get rid of their beta endorphin, they were also making antibodies that were getting rid of, that was blocking the opiate receptors. So they got, in two ways, it screwed up their cellular signaling. And so, I mean, what I want to say on this is, maybe I should say this for the, yeah, I'll save that for the next paper. So, presence of elevated levels of IgM class autoantibodies normally reflects recent antigenic stimulation. So the problem, if your IgM levels are elevated, that means that your immune system is getting acutely or recently activated to raise those levels. So this would be a problem because it would indicate ongoing reactivation of your immune system or a very active autoimmune disease. Anyways, unexpectedly, we found increased levels of hypercrin 1 total immunoglobulin autoantibodies and anti-idiotypic IgM autoantibodies in all three groups of patients, and particularly significant in the hypersomnia group. These results may signify a continuous antigenic stimulation during the disease progression. Findings supported finding supported by a negative correlation noted between disease duration and levels of IgM autoantibodies. So the presence of elevated levels of IgA class of autoantibodies may reflect luminal, most commonly intestinal or respiratory tract origin of antigens. So the IgAs are frequently a result of things that are going on within your digestive tract, or apparently it's also within the respiratory system, although I'm not well educated on that. So we found that narcolepsy with cataplexy and HI patients had increased percentage of immune complexes of hypercretin 1 IgA class of autoantibodies. So this finding, this made signify that class switch from IgM to IgA hypercretin 1 autoantibodies. This switch is triggered by a luminal antigen, so something that is in the digestive tract might be causing the initiation of this snowball effect. So if we could figure out what is the cause of these autoimmune diseases, we can do a better job at removing that antigen that might be aggravating this autoimmune response. So if you imagine, think of it as like celiac disease. Celiac disease, if you're eating gluten, there is an antigen in gluten that some people will make antibodies against. And if they make antibodies against it, then those same antibodies can attack a protein in the body, an enzyme in the body, and that can cause very severe digestive problems, and then also there could be some skin manifestations as well. Anyways, if we could figure out if there was a dietary cause to narcolepsy, or at least aggravating narcolepsy with cataplexy, then that would be great, because then we could tell these people to cut that particular antigen out of their diet, and that might theoretically prevent them from progressing worse and worse, or at least slow it down. Anyways, we previously showed existence of sequence homology between hypercretin 1 peptide and some microorganisms, including gut microflora. So I've done previous videos on this before, but sometimes microorganisms will have amino acid sequences that look very similar to sequences that are within you. And so this is called molecular mimicry. And so those microorganisms, if you produce antibodies against that particular amino acid sequence of that microorganism, that could theoretically affect your metabolism and affect your cellular signaling. It can affect your hormones. So it could be a strategy at which some bacteria utilize in order to evade the immune system and to live happily within your body. So especially since if antibodies are being used to attack your own proteins, then those antibodies aren't free to attack the microorganism. And remember, you have a lot of microorganisms that just live happily within your gut. So it's not necessarily a bad thing or a wrong thing that this is happening, but we need to be cognizant of that molecular mechanism. We need to be cognizant of it, because some microorganisms are using it for something that is mutually beneficial, but a pathogenic bacterium or a pathogenic pathogen microorganism will possibly use that same strategy or that same mechanism to screw you up and cause pathology. And that's what we want to avoid. So this may account for the concept of molecular mimicry responsible for production of cross-reactive autoantibodies. So one other thing that I want to put in is the bacteria, the microorganisms, especially the viruses that circulate around the human population year after year after year. We are somewhat stable with those, especially the pandemic ones. The pandemic ones or the endemic ones, we've co-evolved with them. After a while, we reach an equilibrium state. We reach a new normal. My concern right now is that we are being introduced with a new coronavirus, which has a very large genome, And this very large genome can have a lot of new peptides that might cross-react with something in your body. And I'm more of the belief that they are cross-reacting with something in your body, especially with some of the immune hyperinflammatory responses that we've been seeing in a subset of people who contract the disease. Okay, so what is my concern? Well, one of my concerns is an autoantibody that is that can attack a that can attack a prolactin release inhibiting peptide in the body. There's a good sequence that shows a lot of similarity with that, and so if you make antibodies against that, you might release way too much prolactin. And apparently, if you make an antibody, then you might actually start stimulating, or oh wait, no, if you, sorry, I mixed this up. If you make an antibody against that peptide, you might actually, release too much prolactin. And if you make an antibody, you might start suppressing prolactin, or it might become more and more difficult to regulate your normal prolactin levels because the entire time your body's making more and more antibodies attacking each other, forming these larger complexes, and as they become more and more activated, you become more and more dysregulated because it's harder and harder for your normal signaling to basically cut through the noise, because the noise is getting louder and louder with more antibodies being produced. So that is one of my concerns of a possible long-term consequence of contracting COVID-19, and I hope as few people as possible end up experiencing that. Regardless, let's continue. Interestingly, patients with recent onset of NC displayed elevated anti-streptococcal and tribbles homolog 2 antibodies. So people who were recently pushed over the edge into narcolepsy with cataplexy, they also have elevated anti-streptococcal and tribbles homolog 2 antibodies. So it is possible that streptococcus might be one of the organisms that has molecular mimicry of something in your body, and that might be causing the push into narcolepsy with cataplexy. And the tribbles 2 homolog 2 antibodies, this is another, it's a semi-vestigial enzyme in the body, and perhaps it shows some homology with some parts of streptococcus. I'm not going to go into much detail, but this might be one of the things that pushes people over the edge to cause that snowballing effect. Anyways, disease duration correlated positively with levels of IgA hypocretin autoantibodies, suggesting an increase of luminal antigenic stimulation to produce such autoantibodies during disease progression that, again, may be triggered by yet unidentified molecular mimicry of microorganisms. So although several sleep-related characteristics such as daytime sleepiness, sleep onset latency, and hypnagogic hallucinations were significantly associated with serum levels of hypocretin-1 autoantibodies in patients with central hypersomnia before correction for multiple tests. These associations were not significant after such a correlation, and therefore, functional significance of these correlations should be individually verified in further studies. So, excess daytime sleepiness is a hallmark symptom of primary central hypersomnias. However, the neural basis of sleepiness remains unclear in most disorders. So it's idiopathic. We don't know. We don't know the cause. So based on the present data, the relevance of hypercretin-1 reactive autoantibodies and mechanisms underlying sleepiness may be hypothesized even if the relationship between the free/immune complex system immunoglobulins and the hypothalamic sleep control remain to be clarified. So we still do not know the exact mechanism by which this the sleepiness can be caused. Or wait, we don't know the exact mechanism by which that could cause this excessive sleepiness. We only have biomarkers. We only have these hints every once in a while. But what is truly going to change the game in solving one of these problems is knowing which protein reacts with which protein, reacts with which protein, which causes X, Y, and Z. And only by knowing that mechanism will we fully have a good grasp on how to attack or cure narcolepsy with cataplexy. So this might be one of the steps, one of the mechanisms by which we learn the full chain of events. We may acknowledge several limitations in our study. So first, there was no measurement of CFS or cerebrospinal fluid hypercretin 1 autoantibodies. And second, we did not assay or test serum levels of hypercretin to see their possible correlations with hypercretin autoantibodies. So the first one, they didn't check the cerebrospinal fluid, and that is a loss because there is not a lot of, I should look into this. I don't know if the CSF is before or after the blood-brain barrier, but it would be important to see the antibodies not in the serum of the blood, but see what are the antibody levels closely related to the nervous system, because that's where these receptors and these peptide hormones are being produced in the first place. So if all the autoantibodies are in the blood, not interacting with the nervous system at all, we shouldn't expect there to be much clinical problems as a result. So it was unfortunate that they didn't look into the cerebrospinal fluid. And the second one, they didn't test the serum levels of hypercretin to see the possible correlations with the hypercretin antibodies. So they tested for the antibodies. Why did they not test with the actual peptide that we're talking about? We're talking about hypercretin all the time. It'd be nice to have hypercretin graphed in comparison with the autoantibodies, because that could tell us more. Is it, do people produce even more hypercretin as a result of this, or do they produce, or is there less free hypercretin as well? That would be something that would be nice to know. Anyways, third, patients with the hypersomnia and narcolepsy without cataplexy and the controls, they were not HLA matched to the patients with narcolepsy. So HLA matched, this is a major histocompatibility complex. You don't need to know too much. Just know that, for instance, when you have organs donated, it is better to have your HLAs, or I shouldn't say better, it is a requirement to have your HLAs, which is a protein on the surface of a lot of your cells. These proteins actually take peptides that are within the cell and present them on the surface. And so it's good to be HLA matched, because that means you have your antigen presenting proteins look similar. And so if, as I said, if someone needs an organ donated, you would want them to have the same, to match their HLA. You want to match your HLA with each other, because that can avoid an overaggressive immune response. So unfortunately, they did not do any HLA matching to correct for more variables, because as we said, biology is a noisy field, so you need to correct for as many variables as possible. Anyways, the sera was taken long after duration of disease that has decreased the chance of picking up major alterations of antibody levels with relative overlap, even with between group significant differences. So unfortunately, the samples were taken long after the duration of the disease. So it could be that antibody levels are in constant flux, right? So it is possible that the antibody levels lowered back to a basal or a normal state, and as a result, the significant difference might have been even more significant had they collected the sample right after an event had occurred. So finally, the number of control subjects was relatively small. So this is always a problem in every study. The sample size isn't large enough, right? So in conclusion, we found that subjects with narcolepsy with cataplexy display increased serum levels of hypercretin 1 reactive total IgG autoantibodies And all 3 groups showed increased serum levels of complex-forming hypercredin 1 autoantibodies, and this is most likely with the anti-idiotypic autoantibodies. So further studies should precise whether hypercredin reactive autoantibodies are involved in physiological regulation of hypercredin availability. So they should test whether or not these autoantibodies are regulating or dysregulating hypercredin, and if this is relevant to arousal and sleep wakefulness. So I was going to continue on to this paper, but I think I won't because it's already late and I already lost a whole video on it. So I might do a different video that is only like 10 minutes on it. Just know that vasopressin and oxytocin, autoimmune attacks on those seem to be associated with depression, and the inhibition of releasing cortisol, which is one of the happiness hormones. If you release cortisol, that is usually in result to stress, which tells your body to feel good, to feel better after that stress. So as a result, yeah, I think I'm going to stop here. It's already an hour, so we're fine. So if you have any questions, please leave them down in the comments, and I will leave a link to the paper. Oh, wait, yeah, I'll leave a link to the paper in the description if you want to read it for yourself, because I skipped over the entirety of the body of the paper. So with that, have a good one, and I will see you tomorrow.